اوكي يا جماعه، خلينا نبدا. نرحب بالاخ محمد الكورنا اللي بيسوي في دراسه الدكتوراه في مارتن لوثر يونيفرسيتي الله ويتنبرغ، وان شاء الله راح يعطينا عن شغله للبي اتش دي زي نوع من البروفو للدفاع تبعه للماستر للبي اتش دي ثيسيس. فتفضل يا اخ.
Ready? We are ready? Can you see my screen? Good morning, everybody.
Yes.
First of all, I would like to thank Dr. Mazen Qamsiya for giving me this opportunity to practice my PhD defense. And I would like to thank you, all of you, for attending this presentation. And I am looking forward to hearing from you about the tips and comments and advice, because it will be the first practice talk for preparing my PhD defense. So the title is Many Drivers of the Honeybee Colony Losses in Palestine. So in brief, I will talk about the importance of the honeybees. So the bees and other pollinators pollinate about 1/3 of the food we eat. And secondly, honeybees is considered the major pollinators worldwide, as well as honeybees like supplies the humans with valuable and healthy products such as honey, pollen, beeswax, as well as also the bees maintain the biodiversity and the ecosystems. Additionally, the honeybee is also considered the source of the income for some beekeepers worldwide. Globally, the honeybee is facing colony losses. These colony losses distributed worldwide, and you can see in this map the losses, the high losses reported in the north of the hemisphere, USA, Europe, Middle East, and Japan. And in South Hemisphere, South America, Africa, and Australia, the low losses were reported. So these colony losses were driven by many factors, such as limited floral resources and habitat losses, which, for example, in the USA, they found one major driver is losses of the habitat. As we know, the new developing countries like cutting forest affects on the resources, the food resources of the bees. And the second factor is exposure to pesticides. Also, the intensive agriculture affects on the bees worldwide. And other factors like suboptimal beekeeping management. For example, in Europe, in the last study, the hobbyist beekeepers with small apiaries and little experience in the beekeeping had double winter mortality rate than professional beekeepers. Other important factor is pathogen and parasite, such as varroa mites and other pathogens, bacteria and fungi and viruses of the honeybees are considering the major driver of the colony losses worldwide and causing the dwindling, collapse, and eventually causing the mortality. For example, during the monitoring in Germany, the winter colony losses occurred by varroa mites and some viruses. So now I will go in more details about some pathogens and parasites. First, varroa destructor parasites. Obligate ectoparasites attack all the cast of the bees and is considered the virus vector and occurs almost worldwide, and considering the major drivers of the winter colony losses worldwide and summer as well. And also its effects on the abdomen size of and weight of the adult bees, and also causing the dead larvae. And we see the scattering pattern of the brood. Then also the typical seasonal prevalence of the varroa, as you can see in the map, the red lines, the prevalence of the varroa is low in the spring, then rising to go up in the summer, reaching the peak in the autumn. And also this prevalence depends on the broad development. Next, pathogen, Bacillus larva is pathogenic agent of American foulbrood disease. It's highly contagious bacterial notifiable disease, and also it affects the larva stage of the honeybees. And a few spores will infect and destroy the colony if not treated. As you can see in the photo, the difference between the dead larva and the healthy pupa. And also low losses occurring by these pathogens reported in the Europe and USA. And also 5 genotypes for these pathogens were discovered. And these genotypes like ERIC1 and ERIC2 are more common and are difference in the virulence of against larva. And you can see ERIC1 causing the mortality in 12 days, and the ERIC2 causing faster in 7 days. And also the symptoms of the clinical of this disease normally is very absent, or 90% there's no clinical symptoms. Next, pathogens, Nosema species. Nosema species is small forming obligate intracellular microsporidium of the midgut epithelium cells. And two species of this disease, Nosema abies, in 1909, and essentially in 1996, after 100 years, they discovered the new pathogen, Nosema cerana, and Nosema cerana was more prevalent and predominant worldwide species nowadays. And also, Nosema cerana has a high regional and seasonal variability. And you can see in the photo the difference between the healthy midgut, and the unhealthy. And Nosema cerana, as well as also the difference between the seasonal prevalence, and also we have contradictory result about the virulence of Nosema cerana worldwide. At the individual breed level, you can see here in France, USA, Germany, and Finland, and the mortality was varied between 10 to 40, so it's low to moderate. But in the Spain, the mortality was 100%, so very high. Also at colony level, in the North Europe, the losses from this disease, from this pathogen is low, but are high in the Spain. So now I will go to talk about Palestine. Palestine is located in the East Mediterranean zone, and it characterized as the hot summer, which different completely from the European countries, more cold. And regarding the beekeeping in Palestine, we have around 86,000 beehives owned by 2,000 beekeepers. They produce around 1,250 tons of the honey per year. And the data and the beekeepers in Palestine suffering the colony losses, and these colony losses is not unknown that scientifically reported. Like there's no data about, no research or no data like present about these colony losses. So at these problems, as you can see in the photos, we have colony like disorder and collapse, but we don't know the reason. So at these problems, we addressed or established two main aims for my study. The first questions: what's the occurrence and the prevalence of parasite and pathogens in Palestine? So we are looking for 6 pathogens in my study: varroa destructor, Bacillus larva, Nosema species, and Ascosphaera abies, the pathogenic agent of the chocolate disease. This is a fungal disease, and 15 viruses for the honeybees, and Acarabis woodi causing the calmites. So this is the first questions, and also I am going to looking for determination of the colony losses, because the beekeeper facing colony losses, and we don't know if this is real or not, and what's their drivers for these colony losses. So in order to answer these 2 questions, 2 governorates in West Bank were selected for this study, Hebron and Bethlehem. And in 8 apiaries in this region were selected. In each apiary, 10 randomly colony were selected for this study. And the study conducted from spring 2017 to spring 2019, and 3 seasons per year were visited: spring, summer, and autumn. And during each inspections, We estimate the colony strength, like number of adult bees, brood cells, and as well as we also did photo documentation for each frame. And also during the inspections, we are detection of any clinical symptoms for any disease, and we got honey and bee samples. Then we transfer these samples to Germany for further analysis. So for the first parasite, varroa destructor, we counted all the varroa on the bees by using stereo microscopy. Then we calculated the infestation level by divided the number of the varroa counted by number of the examined bees multiply with 100. And the next pathogen, Bacillus larva, the pathogenic agent of American foul brood disease, we diluted and isolated the honey samples, and we identified the pathogen by cultivation by bacteria agar, special bacteria agar, and then we detected the whitish transparent colony. Then from this suspected colony, we did PCR DNA PCR by specific primers to identify the presence of this disease. Then further for the strain analysis identification, we're using ERIC primers, then DNA sequencing. For the next pathogen, Nosema species, we also did detection by microscope. We examined 30 individual bee colony. So in total, we examined 14,100 individually inspected bees. Then for further analysis like molecular identification, Nosema species determination, we extracted the DNA by phenylchloroform isomer alcohol methods, then using PCR, using specific primers, then DNA sequencing. Now I am going to talk about the results of the three pathogens and parasites. As I said, I will talk in more details about three pathogens, varroa, Bacillus, and Nosema species. First, we will talk about the first question about the prevalence of these pathogens. So the seasonal prevalence of the varroa mite. So this graph, as I told you, we're looking for 2 years and for 3 seasons per each year. So the x-axis is 6 seasons, and the y-axis is varroa mite infestation level. So we did like statistical analysis. We're using linear mixed models because we use this model because we selected some variable as random factors, the colony, the number of the colony, and the apiary, and also we use all the fixed factors. So this model allowed for us to use more statistical analysis in one models. So when we compare and you can see the letters represented the significant differences. And you can see here the first result, the varroa mite in the 2018 is more 5 times than the 2017. And regarding to the seasonal difference, you can see here the highest varroa mite in the summer 18, then low in the fall 18, then very low in the spring 18. And you can see also the same pattern in the 2017, the high in the summer, then getting low in the fall, then very low in the spring, but not significantly. And next to another result, and the beekeepers treated against varroa 2 times per year. The first time before starting the nectar flow in the spring season, then the beekeepers stop treatment during the nectar flow, and then after honey harvesting in the end of the summer, at the end of the July, they treated heavily the second treatment. So they treated 2 times per year. When you go to talk about the next result, the effect of the varroa in the colony population size, as you can see in these both graphs, the y-axis is number of the adult bees, and in the y-axis in the second graph in the capped brood cells, and the x-axis the number of the varroa mites per 100 bees. And you can see very clear the number of the adult bees is high at the zero varroa, around 80,000 the average per colony. Then the number of the adult bees declining sharply, as you can see, until the around the half of the number of the adult bees when the varroa arrive 50. As well as also in the capped brood cells, which, as we know, this pathogens, varroa, attack the brood, mainly brood. And you can see the number of the brood cells also decreasing very sharply to arriving 1,000 brood cells when the varroa reaching 50 varroa. So now we are going to talk about the second pathogen, Bacillus larva. So the seasonal prevalence, we found in the fall 17, the number on the infected colony higher than the summer 17, as well as also in the fall 18, we found higher than summer 18. And the next graph, reflecting the CFU of the bacteria, also reflecting the same pattern of the infected colonies. The number of the CFU is higher in the fall 17 and 18, which this result is very unexpectedly the high prevalence AFB in Palestine. And we going to more details. We are looking for the genotypes in Palestine because, as we know, there's variants of these genotypes. So we determine the ERIC1 and ERIC2. The ERIC1 is yellow, and the ERIC2 is red. Surprisingly, we found the ERIC1 is still more prevalent than ERIC2, but we found ERIC2 in the two apiaries, in the Battir and Al-Dahriya, and we didn't find any genetic transmission routes or what's the reason for this transmission between these ERICs, especially ERIC2, how we got this ERIC2. Because far away from Battir to Al-Dahriya, from the Beit Lahem, and there's a big distance, so we don't know the real reason for this transmission routes. So the third species, Nosema species, so we found also the high prevalence in spring 2017, then low in summer, then small peak in the fall, and also the same in the 2018. We found high in the spring, then low in the summer, then small peak in the fall. And next, the Nosema species determination. So we found similar trend for the global issues. We found the new pathogen Nosema serrana is more prevalent, and in the old samples in Palestine, and we found only Nosema apis, the old species, only co-infected with Nosema serrana. So from this result, it's very clear the Nosema serrana new pathogens is more prevalent in Palestine, and the data about these pathogens worldwide is still new. So we go from this result, we go in the further new result. So we're looking for the genetic population of the Nosema serrana in Palestine. So we amplified the 16S small subunit of the ribosomal DNA of the Nosema serrana in Palestine. So we did cloning for 8 haplotypes. So we found, surprisingly, we find our nucleotides, the red, the blue letters. This is the haplotypes of the Nosema serrana in Palestine. We found this unique haplotypes different from the reference of Greece in the 1996, and also different from the other reference strains worldwide. And you can see here the red italic letter. This nucleotide is only we found in our haplotypes. So that means our haplotypes is unique in Palestine, and even also in more details. And you can see these new clones or new haplotypes we isolated from 1B. So that means even in 1B, we have different sequences. We can say different sequences or different copies of the ribosomal DNA, even within one colony. And you can see the different nucleotides For example, in node 79B1, we have GAATT, which is not present in the reference or in the other haplotypes in the same bee. So from this result also we going to did more details. We did the maximum likelihood trees to see our haplotypes. It's different from global isolates or similar to Palestinian isolate. We found also this like big distance, genetic distance between our samples and also the global isolates. And there's no like consistent pattern for our isolates. And you can see, as mentioned before, one bee has 3 clones. Also, the distance between them very high because they have very high variability of the nucleotides. So, from this result, we found like unique haplotypes in Palestines, as mentioned in the introduction about the contradictory result about virulence of the Nosema serrana. So we did a small experiment in Palestine to see the virulence of Nosema serrana is kill the bees 100%, like in the Spain, because it's very close to us, or doesn't kill the bees. So we did like a bee cages experiment in the Hebrew University. Then the results show us we used the Kaplan-Meier survival curve to show the difference between the infected group and the control group. And as you can see, the blue line is the infected by Nosema serrana, and the red is a control group. And you can see, and the x-axis is the days post-infections, and the y-axis is survival of the bees. And you can see the Nosema in Palestine kill around 30% of the bees, not 100% like Spain, which has been close to Palestinian, which our results support the North Europe that Nosema doesn't kill 100%. Now we will go to the second aims of our study, the annual and seasonal colony losses. So we found unexpectedly annual losses. In 2017, we found 26 colony losses, and in 2018, we found 29% of the losses, which is very high. It's not expected. And we found the losses distributed during the 3 seasons. Half of the losses occur in the winter time, and then follow in the summer, then low in the spring. So the second questions, we want to know the drivers of these colony losses. How happens, which is the driver of these colony losses? So we found first, around 35% of the losses occurred by Bani Bacillus larva, was something new for Palestinian and also the global studies, and followed by varroa destructors, which is still varroa the major problem in Palestine, similar to worldwide. And the GLM models we used, as I told you, we used generalized linear mix model because these models like estimate more variable and fixed factor and random factors, and also estimate if there is repeated measurable results, variables, and also estimate when the data is not like consistent, like binomial, ordinal, quantitative, and random, normal distribution or not normal distribution. So generally mixed models like use general broad use for this studies. And this GLM models revealed that the varroa mite, when the varroa mite increasing, the probability of the survival decrease. As you can see, at the 0 varroa, the survival is 100, but then the mortality decreased until 50. When the varroa around 50, the colony losses died at 0. And then Nosema serrana, we found very little, around only 2% losses by Nosema. And Nosema serrana, as you see in this, our result, we found 72% of the colony infected by low spores number, 1 million per bee. and only one colony infected by 12 million spores per bee died by Nosema serrana, which this colony died by Nosema 2%. And we found also another factors, it's not biotic factors, is weak colony causing like also losses around 10%. This weak colony, we defined that there's no any pathogens and the population size of the bees less than 2 times standard deviation. And also the GLM models reveal that the probability of the survival increasing, as you can see, when the number of the adult bees increasing in the colony. So when the number of the adult bees arriving 13,000, the survival is 100%. Next to other factors, we found also queenless or queen failure also causing around 17% of the losses. And you can see also the queenless losses distributed during spring and the fall season. Number of dead colonies. And then we found also 2% of the colonies by starvations. The bees like no hunger, no food. And also still we found unknown colonies. We don't identify any like reason for these losses. So in summary, are still around 60% of the losses occurred by biotic factors and around 30 losses occurring by beekeeping management. And so still, and this is the result. So now I will go to the next section is discussion. I will talk about the first aims, the prevalence of the pathogens and parasites. As I mentioned, the varroa sector were detected widespread in all apiaries, which similar to the neighbor countries and to worldwide countries, which as you see in the map, this parasites is occurred worldwide and also the major problems nowadays, still the major problems. And also we found, not like surprisingly, the varroa mites negatively correlated with colony strength because this parasites like mainly fed on the fatty tissue body of the adult and also larva, and similar to not all or most of the studies worldwide. Next to the next pathogens, Bacillus larva, this pathogen unexpectedly we found high prevalence in Palestine, 22%, which similar to Jordan 2012, they found also high in the Jordan, and also as well in the USA around 45%. While but not similar to the European countries where they found low prevalence of this pathogens, and also in New Zealand less than 1%. And next, the last pathogens, Nosema species. Also, we found, like other studies, we found Nosema cellular new pathogens more prevalent and occurs in all areas worldwide, similar to the global trend. And also, we found consistent seasonal patterns, high in the spring, low in summer, and small peak in the autumn, which is similar to the Canada in the recently studied, and but not similar to the many studies in the Higgs group in the Spain and the other like countries. And we are going now to show you the second end or second question, the drivers of the coloniolysis. This is show us again the summary of the drivers causing the coloniolysis, and you can see the thick of the red arrow representing the strength of causing coloniolysis to the honeybees colony, and you can see varroa mite is still the major problems worldwide and also in Palestine. So there's nothing new for the varroa. Varroa is still the major problems in Palestine. And Bacillus larva, which is like unexpectedly, this is the new in Palestine, which not similar to any studies worldwide. For example, in the Germany, for during 4 monitoring years, they found only FB causing less than 1% of the losses. And also a queen failure, which not like new, which also reported in the many studies, for example, in Austria, they found the queen age causing the winter colony losses. So now I will go to the outlook. So as you know, as you see in our study, we have we found massive annual colonies around 30%. This result has recommended us to first establish national strategies for continuous colony monitoring and early disease detections. And also we need action plan to limit the spread of the AFB in Palestine, as well as we need establish beekeeper education and training courses for diagnosis, especially AFB. And we for the further studies, we need to understand or to study the transmission of the AFB, especially ERIC2 in Palestine, and we need also to cooperate with the neighbor countries to know how the ERIC2 arrive Palestine by using molecular techniques. And also as well, we need alternative treatments against AFB and viral sector, because we use antibiotics, so we need something alternative, like, for example, essential oils or some plant extracts for control this disease. So at the end, I would like to thank you, not this group, but I want to thank you, Dr. Mazen, and also you for attending my practice talk. I am looking really for your comments and to improve myself before I go into Germany. And thank you again for your listening and for your attention.
يعطيك العافية.
شكرا كثير، يعطيك العافية. وطبعا المجال مفتوح لأي أسئلة عند الحضور، سواء موجودين معنا وجاهيا أو اللي خارجيا. We open it for questions for anybody, whether here inside this room or online with us. Please go ahead, Mr. طالب الحارثي. دكتور طالب.
بدي أسأل سؤالين والله للأستاذ أحمد. I would like to ask two questions to Mr. Ahmed first about the honey bees. Do we have many species of bees that make honey, or only one species? This is one. Second, we have many types or sorts of honey in the market. Do you think there is a fake honey, a lot of fake honey in the market? You heard me?
Yes, of course. Yes, I hear you. Thank you for your questions. This is not my specialized, but I will answer these questions.
Okay.
And I will give example. The first question: how many species are producing honeybees? As I know, maybe somebody will correct me. Also, the specialized. As I know, 9 species producing honeybees, but the major one is the Apis mellifera produce the honeybees. Apis mellifera, the major species, and the Apis mellifera has around 26 subspecies worldwide. So the mainly Apis mellifera, but we have 9 honeybees in total. We produce honey. This is, as I know, it's not more specific in the honey. And the second questions, of course, we have many fake and many cheating honey in Palestine. I want to give you one example. I think you see my screen. Just one example here. Can you see my screen? So here for ERIC2, how we got ERIC2 in Palestine. The ERIC2 of the AFB, the American Fowl Brood Disease, we got this from imported honey, imported honey, cheating honey. Many beekeepers buy the inequality honey to feed their colony, and we found this honey contaminated, probably contaminated by the spores of the ERIC2. And this honey come from China or from, I don't know from where, but yeah, we have a lot of the cheating honey. So yes, you are right. We have many faking and cheating honey.
In your opinion, where can I find guaranteed honey bee? Bee honey. Guaranteed 100 in Palestine. Where? Because in Hebron we have a
جماعة احكوا بالعربي، احنا يعني بتوقع
يعني
ان الجميع احكوا بالعربي. خلاص خلصنا الديفنس يا محمد.
أوكي.
إذا في
احسن.
أورينيا.
ايوه، لانه في بقول لك هذا عسل السدري وهذا عسل مش عارف ايش هذا عسل الربيع، هذا عسل، وفي عسل بيكونوا بطعموه سكر النحل. وين ممكن نلاقي احسن عسل وموثوق، يعني يكون عسل فعلا، لانه والله الواحد ما هو عارف كيف يشتري انواع احسن نوع.
اوكي. يعني انا بدي احكي لك من خبرتي في وزاره الزراعه، في عندنا دائره النحل، بيسووا فحوصات للعسل وبيطلع في جامعه بيرزيت، وبيسووا فحوصات وبيطلعوا كمان شهادات في هذا العسل. ففي موجود عندنا عسل طبيعي موجود. يعني اذا بتروح وزاره الزراعه بتعرف انهم النحالين اللي بيبيعوا عسل طبيعي، يعني موجود عندنا في الشمال، موجود في الجنوب، معاهم شهادات في العسل الطبيعي.
اوكي،
يعني
شكرا
موجود.
شكرا لكم، يعطيكم العافيه. انا المشكله على 12 في عندي كمان زوم، بدي استودعكم، اتمنى لكم يوم سعيد. شكرا شكرا.
حياك الله دكتور، اهلا وسهلا.
الله يبارك فيك.
الثانية أنا عندي سؤال. محمد، أنتوا في دراسات شبيهة بهاي من الإسرائيليين على النحل اللي عندنا من ناحية الهابلوتايبس؟
دكتور مازن صوتك مش واضح، يعني هابط كثير.
بس إذا في دراسات شبيهة بهاي الدراسات من الإسرائيليين خاصة على موضوع الهابلوتايبس الأرك 2 هذا، أو من الشرق الأوسط بصورة عامة.
اوكي، ثانك يو فور ذا نايس كويستشن. راح احكي بالعربي. راح اشوف اوريكم سلايد شو اللي سويناه احنا. للاسف زي ما حكيت، هذا الباثوجين اللي هو نوزيما سيرانا هو باثوجين جديد، اكتشفوه في 1996 وبدأت الدراسات عليه. للأسف اللي اشتغلوا عليه فقط دول قليلة، اللي هي أسبانيا، اللي هي هيجاس جروب، يعني مشهور في أسبانيا، وعمل هابلوتايبس لنوزيما سيرانا تقريبا لأكثر من ريجن من الريبوزوم ار ان اي، لاي جي اس، للكودينج والنون كودينج. فاللي أسبانيا، وبعدين اشتغلوا كمان على هذا الجروب كمان في ألمانيا واليو اس اي. اما في اسرائيل او في الميدل ايست ما حد اشتغل على الهابلوتايبز او على النوزيما سيرانا، لان لسه نيو باثوجين. يعني احنا اول ناس، حتى انا اول واحد بعد الاسبان بسوي كلونينج لونج فراجمنت للريبوزوم ار ان اي. فلسه انا ثاني واحد بعد الاسبان بعد ما اشتغلوا. ممكن السبب لان زي ما شفنا في تضارب في النتائج، لان هذا المرض يعني وجد في بعض العالم انه ما بيسبب كولونيوسيس، وعاده احنا بنعرف الامراض الفطريه بتسببش يعني هذيك اللوسيس للهانيبيز. فممكن العالم ما تطوروا فيها وما راحوا فيه لفيدرال ستاديز لان ما وجدوا ان. بس لكن زي ما فيش دراسات موجوده في اسرائيل. بس سؤال كويس عشان حكيت عن اسرائيل. الأردن اشتغلوا على هذا الموضوع، بس اشتغلوا إن هل هو موجود نوزيما سيرانا أو لا. يعني بس الدراسات الموجودة في المنطقة اللي حوالينا، هل النوزيما سيرانا موجود ولا لا، استخدموا البي سي ار، هل نوزيما سيرانا أو إيبس. هذا الشغل اللي اشتغلوه. ما حدا راح مور ديتيلز اباوت الجينيتيك بوبيليشن او نوزيما سيرانا او الهابلوتايبس. اسرائيل اشتغلوا نفس الدراسه تبعتي اللي هي درسوا للمين درايف اوف ذا كولونيسيس في 2011، ووجدوا ان عندهم النزيمه الفاروا ستيل ذا ميجر بروبلمز، بس ما اشتغلوا على الاي اف بي، ما اشتغلوش على الحضن الامريكي، اشتغلوا بس على الاي اف على الفاروا. وجدوها ان هي على شيء عندهم هي مسببه لانهيار المستعمرات في اسرائيل. هذا هو بس يعني بالنسبه للهابلوتايبس.
إذا ما في ملاحظات ثانية أو أي أسئلة، المحاضرة بنسجل فيها فراح نوديها للجميع حتى يكون عندكم الريكورد. كنت بقول المحاضرة تسجلت ونودي لكم التسجيل. في أي أسئلة ثانية أو أي أحد عنده ملاحظات؟ شايفين عاد الساعة 12. يعطيك العافية. أنا يعني امبرست يعني محمد الكرنا، الله يعطيك العافية. وكنا بدنا نزيد من الدراسات الفاصلية لمشاكلنا ولطبيعتنا والتنوع الحيوي والتنوع الفيروسات والتنوع البكتيريا والشغلات هاي كله مثل دراسة. يعني إن شاء الله يعني لازم تنشرها يعني لما تخلص
ان شاء الله.
يعني أكثر تطلع منها أكثر من بيبر يعني نستنشر بيبر واقف على one species يعني كلها.
هيك النية ان شاء الله، هيك النية ان شاء الله.
يعطيكم الف عافية وشكرا للجميع للحضور. زي ما حكيت نلقيكم السلايد البرزنتيشن وان شاء الله كمان الاسبوع الجاي او اللي بعده يعني. يعني على القليلة كل اسبوعين، بس ممكن يكون كل اسبوع نعمل نشاط زي هذا انه نقوي حالنا ونستفيد. انا شخصيا استفدت كثير من محاضرة اليوم وان شاء الله تساعدنا في المحل اللي عندنا. رامي مساعد محمد نجاج هنا نستفيد من. شكرا للجميع، يعطيكم العافية.
شكرا لكم. شكرا دكتور مازن، يعني دائما دكتور مازن بتحفنا وبسمح لنا نتطوع ونجي نستخدم الانترنت وكل شيء بيعطينا فري، نستخدم بدون مقابل مش زي بعض المناطق ثانية. فيعني بشكرك جدا دكتور مازن على إتاحة لي الفرصة إني أسوي براكتس توك، وشكرا لكم.